I'm going to talk about how MaizeGDB is dealing with our explosion of gene models. And you can interrupt me anytime with questions or suggestions or comments. And then I hope at the end of this short presentation, we can discuss sort of the way forward, because I feel like everybody is dealing with this. So first of all, how did we get so many genomes? We have almost 50. We have 48 on the website, a couple more in the works. We did ask people at the Maize genetics meetings. We decided that we were going to host lots of different genomes so that people could study the diversity in genome biology. We do have, we advertise where information on how you can contribute, well, all types of data, but particularly genome assemblies, and also how to make them fair and all our data fair. And if you want to be what I'm calling quote unquote hosted at MaizeGDB, you have to submit your genome assembly to NCBI or EBI. That's an absolute requirement. We can help you a little bit with that. We've helped a number of groups, but absolutely has to be submitted there. Just the assembly, not necessarily the annotations. You must use our genome nomenclature, and that has helped a lot. And then you must complete our more extensive metadata template than NCBI's, and that has already come in to be very handy. So when we host a genome assembly, for the most important genomes, what we do is we provide metadata that's more complete than you can find on the NCBI project and all of those. We provide a JBrowse instance. We can remap data tracks if it makes sense. We provide blast targets, and then we will add that genome to a pan-genome calculation that Maggie Woodhouse does in-house at MaizeGDB. We don't do this for all the genomes, but currently we do it for most of the genomes. We have different levels of support. So if we eventually get 3 different sequences from one inbred, we may not do all of them. Oh, and actually there's at least a number 5. We do provide downloads of all the information for that. I'll show you that in a second. So we do have one genome that we spend more resources on than others, and that is B73. B73 is the inbred that was first sequenced in 2009, and there are actually 6 versions of that genome available. The V5 version 5 is the current one, and it's stellar. It's very, very good. We call this the representative genome. We sometimes still call it the reference genome. NCBI does this also because in humans, I think there are over 900 different genomes, but NCBI only serves one of those genomes, the representative genome. So most of the genome assemblies that we support are reference quality, and we don't anticipate taking in any more genomes of lower than reference quality. So how do you find the genomes? I'm just going to quickly show you that we have a lot of places you can find it. It's very redundant because you all know that people can't see things right in front of their face. No, I mean, including myself, on other websites. So quick links to JBrowse and GBrowse. We're moving to JBrowse, but our older genomes we're just keeping in GBrowse. And we have information on this really cool set of 26 genomes from the Nested Association Mapping Panel. These are the founder parents that were all intermated with B73 to generate a whole series of recombinant inbred lines, 5,000 lines. These are the 26 parents. They were all sequenced, assembled, and annotated by one group using the same wet lab and in silico pipelines. And so they're directly comparable, and that's a really great thing. We have more information, and we've set up on JBrowse how you can jump from one genome to another, so that there's a lot of cross-pollination there. Downloads, we provide a lot of downloads, but we did make the decision to provide all genome assembly and annotation files in one place with sort of a standard nomenclature to the file names so that you can easily find the CVS annotations or whatever you want for each of the genomes, because it can be hard to find this out in the world. We also have a SciVerse page with this data too. So if you want information on all the genomes, we have a couple places to do that. We have a big button on the front of the page. You can go to the Genomes Data Center here through the drop-down menu on the top. We have a genome drop-down menu where you can go to individual genomes directly, or again, the complete list. So I'm going to show you what the complete list of our genomes looks like. We organize our data in data centers so that we can have special search functions for each of the data types. And this is what the Genome Data Center looks like. Of course, I'm just showing you the tops of the pages. You can search the genomes. We started grouping genomes into sets because we imagine we're just going to get more and more of these. We're not going to continue to host everything, but those decisions are still fluid. And then we have a list of all the genomes that we have here with links to their metadata and NCBI and some information on them. Okay, so if you click on any one of these genomes, you get to that individual genome page. So this is for the line CML228. When you go to an individual genome page, you will land on the browser for that genome. You can see here that this is the name of this genome. But you'll notice that we have these tabs here at the top, and this is now becoming a feature at MaizeGDB because we want to consolidate data on one page, but there's just too much to do it at just one page. So if you look at the project details, that will tell you about whatever project this came from. For example, the NAM parents, those project details are all the same for each of the NAM genomes. So you'll find a lot of information here. And then the metadata is the data that we collect, and we show that. So you can find each of these three things. So now we have 50 genomes. So that means we have up to 50 slightly different gene models for each gene. And then you can, what I'm going to call a gene, I'm going to give you an example of a very well-studied gene that has lots of individual papers on it, was defined by a mutant phenotype, cloned in the 90s. That's a class, we call those classic genes. We have about 500 of them where we have a lot of information. But now we have up to 50 different gene models. They're really technically alleles, but we treat them a little differently than we do phenotypic mutant alleles. So mutant or non-mutant alleles. So for the first 100 years of maize genetics, several thousand genes were identified. Most were by mutant phenotype. And then later, maybe starting in the late 80s, people were finding genes by reverse genetics using sequence homology to genes in other organisms. This is way before any plant genome was created, but there was sequence from, even E. coli or yeast that we could look for conserved genes. So we have a lot of genetic sort of functional information about these genes, like genetic map position, alleles, phenotypes, stocks, all sorts of stuff. However, not all of our genes are yet connected to any gene model. So in order to make that connection, we have to have some evidence that there is a connection. And we don't have that all today. We have about 10,000 genes. Now remember, I'm using gene differently than the word gene model. So we have about 10,000 genes that we have information on them. There's maybe, I don't know, 45,000 gene models of high quality coding genes in any one genome. So we've we got maybe a quarter or 20% of them. They're not all connected to a gene model yet. So when we look in 2009, when we got the first Maize genome assembly, we had genetic information on a single gene, and we had one gene model for that gene. And as I say, we had about 10,000 gene records, and now we have about 100,000 gene models. We had high quality and low quality and different types of gene models. So a little bit more than 45,000, but I'm just estimating about 100,000. So then a few years later, we had the B73 genome had been improved a number of times, and we still had those 10,000 gene records, but now 4 times more gene models. So maybe 400,000. And now today we have 50 genomes. We still have 10,000 genes. Okay, maybe 10,001, but this information takes years to acquire. So this is not changing that much. But now we're up to, 5 million gene models at least. So how do we deal with this? These are some of the issues that we want to deal with. We want to maintain those 10,000 gene model connections that have been done manually through curation. We want to extend this to gene models from other lines, but we want to make sure that people understand this is a calculation. There's not biology, there's not wet lab evidence behind it necessarily. We know that some genes may not have syntelogs in all lines. This could be a real biological thing, or it could be a mistake in anything, annotation or whatever. And there's a lot of genome biology that we don't know. We know in Maize that there are some genes that are in different positions in different lines, but that seems to be pretty rare. If you think about it, we wouldn't have been able to make composite genetic maps over decades if the genes were jumping around and were in different places in different genomes. So anyway, we have a lot of unknowns. And then there's going to be sort of technical difficulties to overcome and this is one example of that. So if you have the gene, the one I'm going to show you is Ligulus 1. So we have this genetic information on this gene, phenotypes, mutants, stuff from papers, all that stuff. And then we have a number of different gene models. So for this gene model, we might have had ontology annotation. Somebody might have provided expression data from RNA-seq in this line, and somebody may have identified a new function in a wet lab in this other line. And so somehow we don't want to have 5 million pages that show that all differently. We want this all to populate up to one place, one PanGene, one gene. So that's one of the issues. Okay, so this is our current approach. This is just what we do now. And then, after this, I would love to have some discussion and hear some thoughts on how other people are doing this and if we could do this in a better way or anything. So what we're going to do is we're going to go to a single gene page. So I worked on Ligulus 1 a long time ago. I tend to use it a lot for examples. We'll go to the Ligulus 1 individual gene record. So this is a very long page, like all of your pages for genes. It has a lot of information. We have, you note the tabs here again at the top of the page. We're on the genetic information page. This tab has images of mutant phenotypes, genetic map coordinates. It has stocks that the stock center provides. It has some ontology terms and the list of the alleles. These are all either mutant or non-mutant alleles, really by phenotypes. So that sort of information is under this genetic information tab. So if we look at the other ones, the other tabs, the gene model is a specific gene model for a specific line. And there's information there. Again, you can scroll down, there's lots of information about this specific gene model, more sequenced centered information. You can go to the genome browser. You can find a lot of calculated information on this particular gene model. You can, we provide the sequence for this exact gene model, the genomic, the cDNA. You know, you scroll down, you've got all the different sequences and their identifiers. And then this is our new tab, the PanGene tab. And this lists all the gene models from all sequence assemblies that are likely to be the same gene based on homology and synteny. So this is a calculated thing. So we want to somehow be clear that this is calculated and not necessarily vetted in the lab. I think we're being super careful here, because if a gene is syntenous and has very high sequence homology, it's probably serving the same function. But we really don't know enough about genome biology yet to really say that for sure. So this is the whole PanGene page for Ligulus 1. And I just want to show you some of the parts that we have. We have a little bit of sort of ever-changing verbiage on our definitions. So for example, we say a PanGene is the set of gene models from multiple genomes that appear to represent the same gene. And we're beefing this part up. We have the analysis description. Maggie Woodhouse does this. It's amazing to me. So she calculated this in, end of last year, and there'll be a brief description on how she did the pangenome calculation. And then, since now she's going to be doing this, when we get a couple more genomes, she'll be redoing this calculation over and over. So each pangenome calculation will be named using a standard nomenclature. So far, it's just MaizeGDB pangenome, the group that did it, and a date. And we'll have associated metadata, which will include the methods used and the genome assemblies used. So yet another entity to begin to have standard nomenclature on is the pangenome results. So then we have the list of all the related gene models in Maize. Now, the first thing to notice here is that I told you we have about 50. I think for this calculation, there's I don't know, there's not all of them were used in this pangenome calculation. So maybe it's 45 genomes, but there's only 32 genome assemblies that have Ligulus 1 syntelogs in them. So that's interesting. Okay, so here's the list here, and you can get to each of those gene models, and you can click around, find lots of information there. Whoops. Then we at the bottom we have the calculated orthologs in other species, and this could get very large, but you can see we have this is all from one assembly, and this is all from a different assembly. So we only have orthologs calculated from 2 different assemblies. These are the sources. We didn't do this calculation. So our current approach to show this, in the actual database and for ease of the user on these tabs, so we got these 4 tabs across here. We have the gene models. The gene model from B73V3 will have its own unique gene model page, which will be connected to its unique sequence page. And that will be connected to one Ligulus 1 PanGene page and one genetic information on Ligulus 1 page. And that's the same for all of these. So if you go to any of these gene models that appear to represent Ligulus 1, they will all be connected to the same PanGene calculation and the same genetic information. So that's how we're doing it right now. We still have a lot of unknowns, and I'm sure this is going to be a little fluid. So yeah, I'm curious how others are doing this and what is most useful for our stakeholders. And let's unmute and talk about this.
Hi, Lisa. This is Tanner.
Hi, Tanner.
That's a great idea. So can you go back to the nomenclature and to show how we will know that a gene, we're talking about the PanGene?
Let's see, was it here?
So
This?
is there, yeah, so is there like a gene model name for a PanGene?
No.
Okay.
That's what we are doing now is, well, let's see, you can,
Can I jump in here?
yes, please.
This is something we've been wrestling with at great considerable length. There is a completely random identifier in the database to hold these together. We've been reluctant to assign formal names to these pangenes because they will change with each calculation. And I'm worried that a gene model may be in it in one calculation and then we improve it and it's no longer in it. So I think to the extent they have a name, it's probably the, if it's a classic gene, the classic gene name. But if it's not, it doesn't actually have an identifier.
We did think it would be great, and this PanGene identifier would be the end-all be-all for nomenclature of genes. It would solve all the problems we have with people in the literature making up new names for the same gene each time they publish on it. But so far, we can't do that. I showed you the case of Ligulus 1, very well-studied classic gene, but there are many, most of the genes are either have little information or no information in this genetic information slot. So those are the ones that make it difficult to have standardization for PanGene names. But, you know, let's work towards that in the future.
Hi, Lisa, this is Sushma.
Hi, Sushma.
Thank you for the great talk, and it's a lot of work. I see that. And so I have a suggestion here.
Uh-huh.
So we have like gene model, we have sequence, and we have PanGene.
Uh-huh.
And then you have here the description about the Ligulus mutant.
Yes.
Something. I thought if you could have like, you know, a tab like gene model, sequence, PanGene, genetic information and function or functional annotation, whatever. Mm-hmm. Perhaps that new tab, when you say function, can get description and reference, and if there is any phenotype known, so that would be perhaps a better organization.
Well, these, I'm just showing you a little bit of these pages. This genetic information tab is very, very long. We have lists of reference. We have all that information. We have different functional information. It's actually a little different. For the genetic information, functional information for Ligulus 1 might be this gene is necessary to provide the periclinal divisions that allow the outgrowth of the ligule structure. That's the function of this gene. But if you go to the gene model tab, you have information on the sequence, and you have a lot of functional information or ontologies from other species that tell you the actual function of the gene. And yeah, all that information is there. It's really spread out between the gene model and genetic information tabs.
Yeah, so my suggestion, I was looking on the website as well. I thought maybe, you know, having it like a function, and then function can be like more discrete functional annotation, and perhaps a phenotype, if there is one for the mutant, could kind of, you know, make it a little bit better. I'm thinking about if somebody is trying to retrieve the genetic interactions and phenotype connections. So I don't know, it may be better to mine that information, that you get just a gene model. You don't, you know, a generic gene, PanGene, and then if you have a function and you have a phenotype associated with it, maybe mining that information to some way may help to build the genetic interactions network or things like that, rather than going into much detailed description and finding information there. So from the machine learning perspective.
Yeah, you'll have to tell me more what you're envisioning, because yeah, that does sound like a good idea, definitely. I don't really know how to do it. How to show that. Yeah.
So yeah, I'm just thinking about the 2 columns, for example, you have like right now 4 columns. So if we have, so the genetic information, when somebody looks at it, so it can be 2 things. So genetic information could be the allele information, it could be the marker information, it could be the SMP information, right?
Yes,
So,
and all of that.
so
Yeah.
intuitively, so not necessarily that people will relate it to a phenotype information. So somebody who is looking for some words, or even a machine, some algorithm is looking into that. So if it may be a good intuitive column that very briefly describes the function and phenotype. So then one can look into the PanGene and then extract the information from the phenotype and the function of the gene, kind of. And so somebody doesn't have to go all the way and dig into the individual portfolio.
Okay, yes, yeah, so that would be like, so if this is 1 page with 4 columns, it would be pulling some of that information out into a briefer column that would, yeah, that would show it all the data, maybe have consistent information across all genes. Is that?
Yeah, I wonder if this might be a job for MaizeMine, where you could make custom queries and pull out just the functional information that you want on a set of genes that you're interested in.
Yeah, that will be wonderful. If there is a, kind of a API or something can be embedded, and you name that PanGene and you, yeah, so something like that will be very nice. And this is a lot of great work. And I'm hoping that how we could go more into developing system level understanding and genetic interactions. So this is what I am thinking.
Yeah, I think that's a good idea. And I suspect that MaizeMine, which is a separate view of the data, may provide at least some of that, and could possibly be extended to do a better job if it's not meeting the needs yet.
Yeah, and I wonder if we could get the genetic information for the genes that we have it in a more formal way. I don't know if all the genetic information is in MaizeMine, because it's
Yeah,
very,
and that's one I'm a little hesitant, because I don't know how much of that's going
Yeah,
on in MaizeMine.
It is not, Lisa.
Oh, okay. Yeah, and I would say we have good genetic information for about 500 genes.
Yeah.
So it's not, whereas gene models, yeah, millions.
If it has coordinates, MaizeMine can probably handle it, but it's a very coordinate-based resource, so just bear that in mind.
And our stakeholders are very used to this. Genetic information tab used to be all that we had for each gene. So people are in the Maize, that use MaizeGDB, are familiar with this this tab.
Because maize is one of the crop that has a lot of mutant phenotype collection as well. We know a lot about the genes and phenotypes. So when you have like the reference 500 genes about which we know a lot, there are a lot of great pictures in literature and mutant characterization. So maybe this is a premier resource that can connect genotype to phenotype very easily.
Yeah, yes.
At least for that set of genes.
Yeah, we'll look into making this this more machine readable, this information.
Hi, Lisa.
Yes.
Yeah, I have a question. So this may have been done like a long time ago, but then how did you initially make the connection between the classical gene to the gene models?
Oh, yeah, that's got to be, well, really, there's a couple different ways that it has been done, but for these 500 genes, there has to be a reference. The 500 genes have been cloned, what we used to call being cloned in the olden days. So we know their sequence, and we know that their sequence is homologous to a gene model. And
Yeah,
we,
so like,
yeah, go ahead.
so the classical gene probably they like before any of the whole genome data came like
Yes,
upon to us, like they may had sequences or something, so you kind of
yes,
blasted the sequences into the whole genome. Yeah,
yes.
and so do you have like for the classical genes, do you have like one single like representative sequence from the reference representative genome or something? Or
I'm going to go to the website. We do call the current version of B73 the representative genome.
Mhm.
And I think we sort of do because we're not making it as easy. This is our nomenclature.
Mhm.
And if you were in the know, you would know that ZM0001 is the B73. This is the 5th version, 2nd annotation of this gene.
Oh, okay. So
So-
that's the name of the gene model of a single representative genome.
Yes,
assembly?
I'm just going to go to the same gene so that we can.
I was wondering if the classical gene has a single representative sequence.
Oh, no, because, let me search this another way here. I'm going to search for Ligulus 1 by gene model here.
Mhm.
And it's still changing and evolving, so it might be different than I expect to be here. But this is now searching all the gene models that are Ligulus 1. However,
Mhm.
the B73 gene models are going to really come up first.
Oh, okay.
So there is a large number of gene models, but it's only the first 100 are shown. So you can, oh wait, there's 4 pages here. Ethy, do we have all, if you search, do you get all the gene models or just the B73 gene models? Ethy and Maggie are just so wonderful at this. I think it's something that our stakeholders want to be able to compare across genomes because the diversity in corn is, it's a known thing. And I think that's why they've really done a stellar job mapping so many different things to so many other different things. So this is our representative genome, the B73 NAM. It was made the same way all the NAM founders were. You can also select all these other genomes here to see, but there is a whole bunch of things mapped across these like structural variations, knobs, and centromeres. There's gene model sets from different, here's V4, V3, and oh God, it's, bins.
Get the PanGenome would be interesting to show
So like, which one should I select? here?
above under annotations.
Oh, annotations. Where is it?
Keep going down.
Oh,
Right
oh,
there.
there it is. That's what I'm looking for. Yes. Okay. Yeah. So I'll put one of these on. Okay. So this is the official gene model is at the top. And then here's the gene model from V4 of the same genome. Here's from V3 from the same genome. But here's the genome lifted over from a different genome, CML22.
So that mapping is done by pangenome analysis, right? Yeah.
No, I think
No?
this particular one was lifted over.
Hmm.
They are calculated a little bit differently.
Mm.
In other words, some of these, we have like 900 tracks on our, I keep wanting to say reference, but it's representative genome B73. We have over 900 tracks. And some of those are mapped directly through a blast-based method. Some of them are lifted over based on coordinate. That they can be done different ways, and that is also different from the pangenome analysis. So not all pipelines are equal.
Yeah, I mean like the under pangenome tab, like on the left side. That might be by PanGenome.
But it says these are lifted.
Oh, okay.
So I think these are lifted over, but these ones might, well, I don't know. If there are comments or other thoughts, or you think of something later, maybe we could continue talking about it on Slack, because it is an emerging issue that we're going to need to be a little fluid about for a while.
Yeah, and getting back to an earlier question, the Maize Nomenclature Committee is quite curious about how to go about naming these. So, you know, if there are ideas, thoughts, whatever, that would be great to get that.
Naming these, you mean pangenome sets?
Yeah, the PanGenes, coming up with PanGene names.
Oh, yes, absolutely. Like Tanner asked, yeah.
Yeah,
Single identifier for a PanGene set.
right.
Yeah.
You know, how do you deal with that when the membership is fluid and the set itself might vanish
Yes, yes.
or be merged?
Great. Well, thank you all for coming. Appreciate it. And yeah, I'd love to know more thoughts about it.
Thank you so